Hello students, today we will be studying the last part of the central dogma, that is the translation. So far we have seen the replication, we have seen the transcription, and today we are going to wind up with that of the translation process, because the nucleic acid has to be translated in the language of amino acid, and that what we have seen it in our last class of the genetic codon formation. Now before we move on to the translation process, we must at first understand the RNA which is responsible for this process of translation. The translation requires an mRNA, side by side it requires a tRNA also, which will be bringing the respective amino acid with respect to the codon which is written in the mRNA, and thus the polypeptide chain formation will take place, resulting into the formation of the protein. So the expression will be in the form of protein, although the information is stored in the form of the nucleic acid. Now this tRNA is also known as the adapter molecule. Now why is it known as the adapter molecule? It was Francis Crick who, even before the knowledge of the genetic code by the various scientists all across the world, proposed that there must be a molecule which can be reading in one side the codon which is present in the mRNA, and with respect to it should be assigning a amino acid which later on will be forming a peptide bonding between each other to result into a formation of protein for the expression of any character. Although it took a lot of years to finally postulate the model of this adapter molecule and how it is associated or how the working is done, but it was said much ahead of its time of the entire structure of this tRNA by Francis Crick. Now this tRNA or the adapter molecule is also known as the soluble molecule. Why it is known as the soluble molecule? Because it is soluble in 1 molar of your NaCl. Okay, so that's why this is also known as your the soluble RNA. And this tRNA or the transfer RNA is peculiar in the sense that it is not formed of so many nucleotides. It is just formed of 100 nucleotides or a few more, and these nucleotides sometimes, because of complementary base pairing, forms a looping structure and forms a kind of structure which appears to be like that of a L alphabet in English, or sometimes it is also known as a clover leaf model. Okay, so this is how the structure of the tRNA looks like because of the complementary base pairing between the that is your nitrogenous bases. Now it has 4 arms, and sometimes in some tRNA additional 5th arm is also there, which is called the variable arm. Now this variable arm's function so far has not been found properly, but the 4 arms which it has, that function is justified or is known to everyone. So let us see what it has. If we see this as a single-stranded single-stranded RNA, in that case it will be having one 3' end and a 5' end. So 3' hydroxyl N is having the site which is called as your amino acid binding site. That means this is the place with which it will be binding with the respective amino acid, which will be with respect to the codon which is present here, because this tRNA will have an anticodon loop which will be having the base sequence which is complementary to the base sequence which is found in case of the mRNA. It is in the form of triplet here. So with respect to these 3 base sequences in the mRNA, these are the anticodons. Suppose this is CCG. So C will be having the complementary base pair G, C with G and G with C. So CCG or GGC. So with this kind of tRNA having the anticodon loop as a GGC will be bringing a respective amino acid with respect to this codon which is present in amino acid. It is not with respect to the anticodon loop, but it is with respect to this that is your mRNA codon, that the amino acid will be decided. Okay, and always there is an initiator tRNA, and it will be always bringing the methionine. Though in sometimes some prokaryotes it is also seen that the starting codon is GUG instead of AUG, which will be coding for that of the valine, but generally it is methionine, and the tRNA is bringing the first tRNA, the initiated tRNA always brings the methionine as the first amino acid to start the translation process with respect to the mRNA. Now, so this is the amino acid binding site. The next is the ribosome binding site. Now, as we know that for the translation of the protein, the mRNA comes to the cytoplasm, cell cytoplasm from the nucleus. So, once it comes to the cell cytoplasm, the ribosomal subunits are actually detached. They are not in their original shape or original form. So, these ribosomal subunits, when the protein synthesis is required, they associate with themselves, the smaller subunit and the larger subunit. Suppose if it is a prokaryotic cell, in that case we know that the ribosome is 70S type. So, if it is 70S type, it will be having the larger subunit and smaller subunit. Larger subunit will be of 50S type and the smaller subunit will be of 30S type. So, the smaller subunit, which is 30S type, it will come and bind with the mRNA, which the structural genes which has to be translated, there it will be binding it. Okay, and now the larger subunit will come and will get associated with the smaller subunit, which is of 50S type. Now, this larger subunit further is made up of various other subunits. It has parts. So, in that there is 23S type of RNA, which is having a catalytic function, which is a enzyme which is called ribosome, and it is also responsible for the peptide bond formation. So, those areas will be created only when the two subunits, the ribosomal subunits, will be joined together. Okay, there will be the place, or there will be the areas which will be formed as a result of association of the smaller subunit with that of the larger subunit, where the amino acid containing the tRNA will come and bind, and where the peptide bond formation will take place, there also one region is there, and another region is also there, which is called the exit region, although it is not mentioned, but in your book only P and A. P means the site where the peptide bond formation takes place, and A is the site where the tRNA containing a new amino acid will come and position itself. So, when the peptide bond is formed between the the two tRNAs, one in the P site and one in the A site, then P site tRNA is released from that of the amino acid, because it has given its amino acid to the new amino acid brought by the tRNA in the A site. So, as a result, since the peptide bond formation has taken place, now this tRNA which will be in the P site is free. Now, it will shift to another site which is called the E site. Now, E site is exit site, and from there it will be released out, because it has donated, or it has given its amino acid to the polypeptide chain. So, this way the movement or the shifting or the reading of the codon which is present in the mRNA will be done by the ribosomal subunits. Now, so this was the ribosome binding site. So, this tRNA which will be bringing amino acid will bind with the ribosomal subunit by the help of this arm, which is called the ribosomal binding site. And this is the third arm, which is the most important arm, that is called the Anticodon Loop. It will be having the complementary basis, the complementary codon with respect to the codon which is present in the mRNA. So, whatever codon will be here, with respect to this only, the tRNA which can form, or which can form a bonding, complementary bonding with this, only they will be coming and binding with that of this codon present in the mRNA. And this fourth loop is the DHU loop, that is dihydrouracil loop, and this is the loop which is actually amino acid activating synthetase enzyme binding site. That means with the help of this loop only, the synthesis of the polypeptide chain will be taking place. So, this will be the enzyme, it will be binding with the enzyme portion of that of the ribosome, which will be responsible for the formation of the polypeptide chain. So, all these loops or all these arms of the tRNA or the adapter molecule is very important in the process of translation. This one will bring the amino acid, This one is the anticodon loop, which will be complementary with respect to the base pairs present in the mRNA. This one is the ribosomal binding site, that means it will be going and binding with the respective position of the ribosome. And then this one is a DHU loop, that means as a result of this DHU loop binding with that of the ribosomal subunit, that activation of the amino acid synthetase enzyme will take place, resulting into the formation of the polypeptide chain. So this is all about your adapter molecule or the soluble molecule, that is which is called as your tRNA. Now how does the process of translation takes place? Translation process takes place by the help of, I told you what things are required, that is one mRNA is required, which of course is formed from a double-stranded DNA, and then the tRNA is required, and the ribosomes are required, which is very important. Now the ribosomes are formed of what? The ribosomes are formed of again RNA and some special type of proteins. Almost 80 proteins are involved in the formation of the ribosomal subunits, and the association of the two ribosomal subunits also takes place in presence of the magnesium. So these are the various things which are essential for the process of the formation or translation of protein from that of the nucleic acid. Okay, so when the initiation starts, there is an initiator codon, and when the termination ends, there is a termination codon, and in between is a structural gene which has to be translated. Now every time a translation takes place, there are two regions, just before the starting and just after the end, so that the translation process is regulated properly, so that there is no disturbance in between the formation of the polypeptide chain, and they are not acted upon by any other catalytic enzyme. So this is also regulation. Now this regulation is at the translational level. So this is again a regulation of the gene at translational level. So regulation of the gene takes place at various level. At translation level, what is the regulation taking place? That is just before the start codon and just after the stop codon, there are two flanks of certain nitrogenous base pairs which does not code for any kind of protein. Okay, now the start codon, we know initially the start codon is AUG, and with respect to it, the adapter molecule brings amino acid which is methionine. So it will come, and at first it will bind with that of the that is your A site. Okay, now next, when it will be moving, the ribosomal subunit will be reading the sequences over here. So it will be coming from the A site to that of the P site. So when it is coming, before that, one more thing I forgot to tell you, that activation of the amino acid has to take place. Now this activation of the amino acid, because it has to bind with that of the tRNA, so it has to take place in presence of ATP. Okay, so that is the important step for the process of translation. So at first, activation of the amino acid with that of the tRNA has to take place in presence of the ATP. Then only because for the formation of peptide bond, energy is required, so that energy will be given by that of the ATP when the two respective amino acids will lie side by side, and there will be an enzyme, and that synthetase enzyme, amino acid synthetase enzyme, will be activated only in presence of the enzyme. So the next tRNA with respect to the next codon present in the mRNA will bring another amino acid. So when this amino acid will come, it will come at the A site of the ribosomal larger ribosomal subunit, and when it will be coming and lying close to the amino acid which is in the P site, so as a result of the ATP, what will happen is that also in presence of that enzyme amino acid synthetase, there will be a peptide bond formation from here to here. Okay, you can see the arrow. So from here to here, the peptide bond formation will take place. So as soon as the peptide bond formation take place, now this tRNA will move to this next position, and this tRNA will come to this E position, and it is released from the that is your ribosomal subunit, because it has donated its amino acid. So this way the third tRNA will come over here in the A site, and by this time it is having two amino acids. So third one will be coming. So since they are lying again close in presence of that enzyme, again in presence of ATP, the third amino acid also will form the peptide bond. So this way a long chain polypeptide will be formed till it reaches the termination codon. So once it reaches to the termination codon, there are some termination factors. Now those termination factors along with that of the termination codon is responsible for the release of this polypeptide chain from this that is the tRNA, the last tRNA, and for the stop codon there is no tRNA, there is no amino acid. Okay, and once this polypeptide chain is now released, then the two ribosomal subunits again get dissociated. Okay, they get dissociated, and now this polypeptide chain which is formed, this can be used variously. It can be used directly, or sometimes it can be a part of the endomembrane system. It can enter into the endoplasmic reticulum, it can undergo some processing, it can take part in the membrane biogenesis. Even it can through the endoplasmic reticulum, it can move on to that of your Golgi bodies, and there the processing of this polypeptide can take place, resulting into the formation of a full-fledged enzyme or that of the hormone to be utilized by the cell. So this is how the process of translation takes place in case of a cell, starting from that of the replication till the formation of the mRNA, that is from any one strand of the DNA, of course, the template strand, which is from 3' to 5' direction polarity, and then finally with the help of the ribosomal subunits, with the help of ATP, with the help of the tRNA, the synthesis of the polypeptide takes place. So this is about all the things so far being discussed, and then next is of course the regulation of genes, which we will be seeing in the form of the lac operon. That's all for today's class.